Yeah, thank you. Yeah, so hi, good evening, everyone. I'm honored to be here today to present a topic on quality control of cell therapy products from the perspective of EU ATMP quality regulation. So in today's presentation, I'll bring you through the definition of ATMP, how EMA and EC regulates ATMP in the EU, followed by using the manufacturing process of CAR T cells therapy as an example to illustrate how the quality of ATMP is being regulated. And to bring us closer to home, in the last part of my presentation, I'll briefly bring you through how cell, tissue, and gene therapy products are regulated in Singapore. So without further ado, let me introduce on ATMPs. So in EU, medicines based on genes, tissue, or cells are termed as advanced therapy medicinal products, in short, ATMPs. The EMA has classified ATMPs under three main types: gene therapy medicines, CTM, somatic cell therapy medicines, SCTM, and tissue engineered medicine, TEM. The definitions of each type, as according to directive, are as follows. GTM contain genes that lead to a therapeutic, prophylactic, or diagnostic effect. They work by inserting recombinant genes into the body, usually to treat a variety of diseases. SCTM contain tissues or cells that have been manipulated to change their biological characteristics, or cell or tissue not intended to be used for the same essential functions in the body. They can be used to cure, diagnose, or prevent diseases. TEM contains cells or tissue that have been modified so they can be used to repair, regenerate, or replace human tissue. As of date, EMA has approved 24 ATMP, out of which there are like 16 GTMPs, 4 SCTMPs, and 4 TETs. The tables below are some of the examples for each categories. So what are the roles of EMA and EC in ATMPs? ATMPs are authorized centrally by the EMA prior to being sold in the commercial market. EMA continues to monitor the safety and efficacy of advanced therapy medicines after they are approved and marketed. EMA also gives scientific support to drug developers to help them design pharmacovigilance and risk management systems used to monitor the safety of these medicines. The benefit of the centralized procedure is that drug developer only need to submit a single marketing authorization application to EMA for review. Once the application is approved, the medicine is authorized to be marketed in all the EU countries at the same time. The blue box on the right shows the other medicines that are approved by centralized procedure. Human medicines containing a new active substance to treat certain diseases, medicines derived from biotechnology processes, advanced therapy medicines, orphan medicines. This is the reviewing pathway an ATMP application will follow upon submission by the drug developer. Both the CAT and CHMP are part of the EMA. The application is first reviewed by the CAT. CAT is responsible for preparing a draft opinion for the quality, safety, and efficacy of the ATMPs. This opinion is sent to the CHMP, and based on the CAT opinion, the CHMP adopts a recommendation on the granting, refusal, variation, suspension, or revocation of a marketing authorization. The recommendation is then sent to the EC for a decision on the marketing authorization. This decision is issued within 67 days of recipient of CHMP's recommendation. So I'll be pausing here now for a poll, which will be very simple. The poll, please. So in EU, who are involved in the regulation of ATMPs? EMA only, EC only, or both EMA and EC? So Christina, should I review the answer now?
Yes, so actually I didn't get any any answers at all. So are
Okay,
you
sure, no
able to
problem.
see my poll? Is everyone able to see my poll?
Yes.
Okay. Well,
So,
okay, sorry. There were no answers.
Wei Xia, can you see the answers?
I
There were
can't actually. But it's all right.
Okay.
The answer is both EMA and EC.
Okay, so
So
78% voted for EMA and EC, 6% voted for EC only, and 17% voted for EMA only.
yeah,
But
so actually
78% got it.
both this EMA and EC are involved in regulation of the ATMPs. Yeah. So CAT and CHMP are part of the EMA. So CAT will draft the opinion as shown in my slide. CHMP will give the final opinion, and then EU, EC will be the one who make the decision upon reviewing the opinion of the EMA. So let me move on to my next slides. So I'll be going through CTD, common technical documents. So how do drug developer present non-clinical, clinical, and quality data of ATMP to EMA? This slide shows the format for the preparation of a well-structured common technical document, CTD, to be involved for the registration of pharmaceuticals for human use. A common format for the technical documentation will significantly reduce the time and resources needed to compile the applications for registration of human pharmaceutical, as well as facilitating the regulatory reviews and communication between the applicant and the regulatory authorities. So this CTD format is also recognized by US FDA authority. So this will greatly save the dossier preparation time for drug owner who wishes to commercialize their drug in the US market. The CTD is organized into 5 modules. Module 1 consists of administrative information and prescriptive information. Module 2 is the CTD summary. So it should contain 7 sections in the following order: CTD table of contents, introduction, quality overall summary, non-clinical overall clinical overall, non-clinical written and tabulated summary, and lastly, the clinical summary. So module 3 contains detailed information on the quality aspect of the drug. Module 4 contains the non-clinical study reports relevant to the drug. Module 5 contains human study reports and related information of the drug. So since today's topic is on the manufacturing aspect of ATMPs, I will be focusing on the modules related to the quality. So the purpose of the quality dossier is to describe the manufacturer's commitment to execute manufacturing and testing of the drug product. It contains information including the manufacturing process and control, product safety, identity, quality, purity, and strength of the drug product. So the objective is to ensure that the manufacturer can consistently produce, analyze, and store the drug product in appropriate standards that's suitable for administration to humans. So the quality information are presented mainly in Module 2 and 3 of the CTD. The QoS of Module 2 is a summary that follows the scope and the outline of the body of data in Module 3. The QoS should not include information, data, or justification that was not already included in the Module 3 or in other parts of the CTD. The QoS should include sufficient information from each section to provide the quality reviewer with an overview of Module 3. The QoS should also emphasize critical key parameters of the product and provide, for instance, justification in cases where guidelines were not followed. The QoS should include a discussion of key issues that integrates information from section in the quality module and supporting information from other modules, including cross-referencing the volume and page number in other modules. The extreme right panel illustrates the content of a typical QoS. So Module 3 contains details of the manufacturing process and quality control strategy. The subsequent images are sections that applicants should include in Module 3. So the bulk of the content will be on 2.3.S, which contains information on the drug substance, and 2.3.P, which contains information on the drug product. The content of this section should include relevant information described in existing ICH guideline. So a unique thing about ATMPs is some ATMPs require continuous manufacturing process due to its biological properties, such as short shelf life at the drug substance stage. This means that drug substance is made into drug product without any interruptions or pauses. So for such cases, Module 3 sections related to drug substance S4 to S7 will not be required and can be omitted. So what are the challenges in quality control of ATMPs? The quality risk associated with ATMPs are highly dependent on the biological characteristic and origin of the cells and tissues. The biological characteristic of the vector, example, replication, competent or reverse transcriptions, and transgenes, the level and characteristic of the expressed protein for gene therapy products, the properties of other non-cellular components, such as raw materials, and the manufacturing process. So in this slide, I will go through some of the challenges that manufacturer might face. So the complex manufacturing process bring challenges in ensuring product consistency from batch to batch. When making, designing, and implementing cell and gene therapy medicinal manufacturing processes, manufacturers need a process to identify the specific risk associated with the product and the manufacturing process and implement appropriate controls. So this risk assessments should consider the significant risk posed by autologous or allogeneic therapies. So for example, for autologous products, where each batch is unique and irreplaceable, the manufacturer must implement enough control to ensure that each batch is of an appropriate quality, despite the limited batch sizes, inherent variability of starting material, and manufacturing process constraint. Supply chain complexity. So most ATMPs are patient-personalized medicines, and the complexity of their supply chain is unique and involve various parties, including hospital to procure the patient's or donor cells, couriers to transport from procurement sites to manufacturing sites to testing labs and administration sites. Thus, traceability is very critical in order to trace back and identify the causes when quality of the product is unmet or when recipients suffered from a reaction after ATMP administration. There must be a documented system to ensure the identification of the tissue and cell during any step from procurement through processing, testing, and storage to distribution to the recipient or disposal. This also implies the ability to identify the donor and the tissue establishment or the manufacturing facility receiving, processing, or storing the tissue cells, and the ability to identify the recipients at the medical facility, applying the tissue cells to the recipients. The traceability data should also cover the ability to locate and identify all relevant data relevant to the products and materials coming into contact with these tissue cells. Storage, logistic complexity. So most ATMPs have short product shelf life and are sensitive to temperature fluctuation. So for instance, changes in storage temperature often will affect the variability and cell count of the cell therapy products, and in turn, affect the quality and efficacy of the drug product. A humidity exclusion in the warehouse may not impact these products directly, but it can damage the packaging seal, rendering them non-sterile. Thus, it is important to develop new shipping, preservation, and quality control solutions, such as to ensure the storage and shipping conditions are properly tracked, assessed, verified, and controlled at all stages of the manufacturing process. So for the third point, the quality of the starting material received from individuals vary from person to person and with the collection method. Product quality may be affected when the starting cells collected are of poor growth kinetics and of reduced variability due to the exposure of conventional cancer treatments. So raw materials such as cytokine can have a significant impact on cell behavior and batch quality. So manufacturer needs to understand the impact that each component can have on the quality of their finished product and develop a system of control to ensure batch-to-batch consistency. Comparability studies are required to demonstrate that there is no adverse impact on the quality, efficacy, and/or safety profile of the product when a manufacturing process change or transfer is introduced to the drug substance or drug product. In some cases, an additional clinical trial may be required to satisfy the health agency. So, as majority of the ATMPs cannot be terminally sterilized, manufacturing process can only be controlled aseptically to prevent microbial contamination. Validation of the aseptic processing must be performed and should include a process simulation test, determining the appropriate release specification. The list of specification tests often depend on the type of cell and gene therapy products and the amount of material available for testing. It is important to identify the appropriate testing assays that can assess the characteristics or attributes of the drug that are mostly important for reliable assessing clinical effect and patient safety. So quality consideration, which I'll be using CAR T therapy as an example. So in the next few slides, I'll be using CAR T cells to illustrate the quality consideration during the manufacturing of CAR T cells. So what is CAR T? It is a type of treatment in which a patient T cells are changed in the lab so they can attack cancer cells. T cells are taken from a patient blood. Then the gene for a special receptor that binds to a certain protein on the patient's cancer cells is added to the T cells in the lab. The special receptor is called the Chimeric Antigen Receptor, CAR in short. Large number of CAR T cells are grown in the lab and given to the patient by infusion. CAR T cell therapy is used to treat certain blood cancer, and it is being studied in treatment of other types of cancer. A typical manufacturing process of CAR T cells begins with the collection of non-mobilized blood mononuclear cells from a patient by leukopharmacies. So this material is then cryopreserved within 24 hours after collection and stored below -120 degrees Celsius. The cryopreserved material is then shipped to the manufacturing facility and stored below -120 degrees Celsius until it is ready for further processing. So upon availability of a manufacturing slot, the patient leukopharesis material is thawed under controlled conditions, followed by cell wash to remove the cryo media. The T cells are enriched, selected, and activated using anti-CD328 antibody-coated paramedical magnetic beads, followed by transduction with a cell inactivating lentiviral vector containing the CAR transgene. Following transduction, the excess vector and other residuals are washed from the culture, and the cells are expanded in static cultures and then in bioreactors. Cell expansion continues ex vivo until there are sufficient cells to meet the final product dose requirement. For cell harvest, the transduced T cells are isolated by separating them from the beads, washed, formulated in infusion media, transferred into the infusion bags, and cryopreserved. Testing of the critical quality attributes of CAR T cells is performed, and after final product release, cryopreserved CAR T cells is shipped to the treatment site. So once patient is ready to receive the CAR T cells, the cells are thawed and immediately infused into the same patient who provide the cells. So the manufacturing of CAR T cells will go through the following major steps: apheresis, cell isolation, cell activation, gene transfer, cells expansion, cells harvest, cells formulation, cryopreservation, logistic, and reformulation. At each manufacturing steps, I will describe some of the main quality factors that have an impact on the quality of the drug product. drug manufacturer can consider this point when designing your manufacturing process and preparing the quality dossier. So CAR-T manufacturing begins with apheresis. In the EU, the donation, procurement, and the testing of the human tissue cells and blood-derived cells should be carried out in accordance with Directive 2004/23/EC and Directive 2002/98/EC respectively. ATMP are characterized by a great heterogeneity. Apheresis product variability is cited as one of the major reasons ATMPs failed manufacturing runs. So most often, this is due to not hitting target therapeutic cell use needed to ensure product potency. Therefore, it is of high importance to create a standardized and robust apheresis collection method. Pharmaceutical companies should do their best to harmonize procurement procedures across clinical sites to minimize the variation in apheresis collection method. Proper cell collection methods should be documented and followed. These documents should be kept for inspection. As for the quality and quantity of starting cells will affect the subsequent manufacturing process and the quality and the safety of the final product. So manufacturers should understand the extent of these changes and implement methods to minimize variability. The manufacturer should have the health check of the cell source, disease detection of the cell source, and the qualification standard records of the cell source. EMA emphasized the establishment of an appropriate testing system to screen for infectious diseases and must assess the risk of contamination of raw material in the supply chain, with particularly emphasis on viral and microbiological safety and transmissional spongiform and septical, in short TSE. In some cases, the quality and quantity of the cells collected from a patient may be affected by, for example, previous treatment received or the patient's current health status. So manufacturers should consider defining patient donor selection criteria or defining characteristics of starting cells that are relevant to producing a product with consistent quality characteristics. The manufacturer should ensure that the following data is retained for a minimum of 30 years after the expiry date of the product. Donation identification code received from the tissue establishment, blood establishment. Internal code or other identification system that is generated by the manufacturer to unequivocally identify the tissue cells used as starting material throughout the entire manufacturing process up to the point of batch release. The manufacturer must ensure that the link between the internal code and the donation identification code can always be established. The cell structural integrity may be at risk due to poor freezing and thawing methods. The freeze and thaw profile should be established for consistent cell quality. The process parameter should be documented and retained, including any process deviation. Cellular starting materials are sensitive to shipping conditions and temperature excursion. Transport and storage procedure and temperature conditions from the point of collection to the point of manufacture should be closely monitored, and procurement should be in place for timely notification of any deviation. Temperature records and deviation records should be kept for traceability. So for the last point, equipment records and supporting data, if applicable, should exist to demonstrate equipment performance. The manufacturing process, all steps after the apheresis, should be performed under GMP and described in detail. In the MAA dossier, the applicant should provide evidence that the process is consistent and the differences in the final product characteristics are caused only by its origin. You may refer to GMP Directive 2003/94/EC, laying down the principles and guidelines of GMP in respect of medicinal products for human use and investigational medicinal products for human use. So the choice of method on cell isolation, enrichment, and activation of cell culture may affect the functional properties of the cell. So once the method for cell isolation, enrichment, and activation are tested and verified and confirmed, the processes are to be followed. Some of the parameters to monitor are the cell purity, percentage of recovery of desired cell population, variability, preservation of cell function throughout the cell culture, and efficacy of this method, which in link to production costs. Manufacturers should consider setting in-process control parameter and acceptance criteria. As cells are sensitive to environment changes, manufacturers need to establish a suitable holding time and storage conditions to ensure the quality of the cell are maintained throughout the duration as well as not to affect the downstream process. So when seed lots and cell banks, including master and working generations, are used, they should be established under the principle of GMP and ICH Q5D. Cell bank safety testing and characteristics are important for batch-to-batch consistency and to prevent contamination with adventurous agents. Viral seed and/or cell banks are to be well characterized in terms of identity, genetic stability, microbial, and viral safety. So for viral vector, there should be full detailed descriptions of the development, genetic identity, and integrity and its microbiological safety. Storage condition and duration may affect the ability of virus to transduce cells. So multiple freeze-thaw cycle, thermal flotation, type of freezing media use, and storage duration may affect the stability of viral vectors. Seed lots and cell banks should be stored and used in such a way as to minimize the risk of contamination. Control measures for the storage of different seed cells in the same area or equipment should prevent mix-up and take account the infectious nature of the material to prevent cross-contamination. It is also important to conduct stability study to ensure that desired quality attributes are maintained throughout the cell shelf life. An optimum viral transduction is achieved when there is a high transduction efficacy, high cell variability, and transduction has low impact to the property of the cell. Thus, it is important to identify control parameters and set acceptance criteria to optimize viral transduction. So the duration of cell culture may affect the functional properties of target cells or expansion of unwanted cell population. So for example, T cells cultured for extended periods of at undesired condition may result in a high number of exhausted T cells lost their ability to kill cancer cells. As such, manufacturers should establish a protocol that allow the expansion of target cells population to a desired number as well as minimizing the unwanted cell population. So for all these 4 steps shaded in blue, these are the quality consideration. So as far as possible, raw material and reagent used in the manufacture of ATMPs should be controlled according to Farm Euro 5.2.12 and relevant guidelines. The use of reagent authorized as medicinal products is preferred. In cases where only materials of research grade are available, ATMP manufacturer is responsible for the quality of the materials. The risk of using research grade materials should be understood. Additionally, the suitability of such raw materials for the intended use should be assured, including where appropriate by means of testing, example through functional tests, safety tests. Manufacturers should verify compliance of the supplier's materials with the agreed specification. The starting and raw material should be free of microbial contamination. The risk of contamination of the raw materials of biological origin during their passage along the supply chain must be assessed. So for the use of bovine serum, it must be certified with certificate of suitability issued by EDQM. So ATMPs are sensitive to changes in their manufacturing conditions. Changes throughout the development phases are often necessary. Changes include renewal of cell lots for production, modifications in the manufacturing process, changes of process scale, changes of a raw material supplier, or additional of manufacturing site sharing the same manufacture process. Any change in the manufacturing process required comparability studies to demonstrate that no adverse impact on the quality, efficacy, and/or safety profile of a product has occurred when a manufacturing process change or transfer is introduced to the drug substance or product. The extent of studies depends on the introduced and the phase of development. Traceability. According to GMP guidelines, a system ensuring traceability of starting and raw materials must be in place. Companies that make ATMPs must be able to trace all their products from where they are manufactured to the hospital or establishment where they are delivered to patients. Hospitals are also required to be able to trace all patients that receive them. Single-use technology. So contamination is a huge risk in the production of ATMP. In the latest revision of EU GMP Annex 1, it states the requirement of manufacturer to increase contamination control measures, barrier technology, and automation to reduce risk to patients and operators. Adopting closed manufacturing system and single-use technology will reduce concern on non-sterility. Notwithstanding this, manufacturers are required to perform biocompatibility studies to ensure the single-use system is comparable with cell culture, with any downstream product and process conditions, as well as identifying and minimizing potential leachable and extractable that can migrate from plastic materials into the production stream. So what are the quality considerations for the last 5 steps of the manufacturing process? A clear batch identification system should be adopted to facilitate the complete traceability to a cell donor. Clear definition of cell preparation batch from cell sourcing to labeling of the final container should be provided. Each stage of the process should be clearly documented. The cell product should be subjected to quality control and lot release testing. The suitability of the release testing and if the tests are able to conclude the quality and efficacy of ATMP are always the topic of discussion. So sometimes in vitro assays are difficult to translate to the efficacy observed in human body. The use of vivo assay as release testing are hard as well. And there are limited pharmacopoeia guidelines on efficacy testing, as it depends on the biological properties of the ATMP. Thus, the EMA places importance to assess the specification data generated from the selected analytical methods and the justification for suitability submitted by the manufacturer. Manufacturers should set the parameters and analytical methods based on the data, knowledge, production, experience, and process validation. Suitability of the analytical methods and validation of key assays should be performed. Acceptance criteria limits should take into account safety and efficacy consideration. It is recommended that an inventory of the retained samples be kept for future analysis. So coming to my last slide of the quality consideration, the final product should be assigned a valid shelf life and in-use shelf life supported by experimental data with regard to the maintenance of cell integrity and product stability during the defined period of validity. Methods for freezing and thawing materials and performing contamination and variability testing should be documented. Transport conditions is a critical impact on the quality of cell product therapy. The adequate of the defined transport condition, example, temperature, type of container, holding time, should be demonstrated. Manufacturers should also ensure that patients receive ATMPs that meet the release specification. However, there may be cases where the administration of out of specification ATMPs is deemed necessary by the treating doctor or when out of specification results are only known after infusion. Drug developers should present in their dossier the potential risk and mitigation strategy to ensure patient safety in the event of out of specification results. And when such cases happen, the drug developer should provide the treating doctors and the relevant competent authority with its evaluation of the risk and how it will be managed. Manufacturers should always investigate the root cause of the OOS result, evaluate the risk, and implement effective corrective actions. So before I move on to my last topic, let us have a short poll again. So in EU, at least how long should traceability data of ATMP be retained after the expiry date of the product? Is it 20 years, 30 years, or 40 years? So I got the poll results, and a majority of you guys got it right. It's 30 years. So as I mentioned in my first few slides of quality consideration, applicants should retain the 30 years of the data even after the expiry of the drug product. So let us come to Singapore. Who regulates cell, tissue, and gene therapy products in Singapore then? The registration, import, manufacture, and supply of cell, tissue, and gene therapy products in Singapore are regulated by Health Sciences Authority, HSA. So in Singapore, cell, tissue, or gene therapy products, in short, CTGTP, is regulated by HSA under the Health Products Act and its regulations, including Health Products, Cell, Tissue, and Gene Therapy Products Regulation 2021. CTGTP are defined as health products intended for use in humans for a therapeutic, preventive, palliative, or diagnostic purposes. CTGTP manufacturers, importers, registrars, and/or suppliers are required to fulfill the relevant duties and obligations stipulated in the Health Product, Cell, Tissue, and Gene Therapy Products Regulations 2021. So in Singapore, CTGTP are risk stratified into two classes below. Class 1 CTGTP and Class 2 CTGTP, depending on the risk of the product. So for Class 1 CTGTP are for products of a lower risk that satisfies all of the following criteria: minimally manipulated, intended for homologous use, and not combined or used in conjunction with therapeutic products or medical device. So for products that do not fulfill any of the criteria, they will be classified as Class 2 CTGTP, which is of a higher risk product. So for Class 1 CTGTP, suppliers must notify HSA on the product and receive HSA returned acceptance of notification before the product can be supplied in Singapore. The supplier will also be required to ensure that the product was sourced from an accredited licensed facility and that it is free from infectious agents. To register Class 2 CTGTPs, applicant must submit application dossier, which are to be written in English and in either ICH CTD or ASEAN CTD format. There are 3 application types to register a Class 2 CTGTP: NDA 1, NDA 2, and NDA 3. The conditions are listed in the right table. Each product registration is done by a specific evaluation route, either the full or a bridge route. The evaluation route will determine the duration needed for the authority to review, the fees incurred, and the documents required for registration. Sponsor must register their CTGTP by the full evaluation route if the product has not been approved by any of the comparable overseas regulator at the time of submission. So for full submission route, sponsor will need to notify HSA at least 2 months before submission of the full evaluation application. Sponsor can also request for a pre-market consultation if it is necessary to discuss specific areas related to their application, including the need for an overseas good clinical practice inspection where relevant. If the product has been approved by at least one of the comparable overseas regulators at the time of submission, sponsor can register the CTGTP via the abridged evaluation route. If you are unsure about which application types or which evaluation route your product falls under, you may contact HSA for advice. So the comparable overseas regulators recognized by HSA are TGA Australia, Health Canada, FDA USA, EMA, and MHRA in the UK. You may refer to HSA CTGTP website for the list of useful guidance documents, relevant forms, and templates to help you meet the regulatory requirements. I highly recommend to read Appendix 8 of the guidance document titled Chemistry, Manufacturing and Control, CMC, Requirements for Cell Tissue or Gene Therapy Products for Clinical Trials and Product Registration. It provides a very useful and detailed information on what is included and should be included in your quality dossier. Companies that wish to conduct clinical trials using Class 2 CTGTP will also require approval from HSA. The trials are regulated under the Health Product Act and Health Product Clinical Trial Regulation. HSA website also provides a list of regulatory guidance to guide companies on how to get their trials approved in Singapore. I strongly recommend companies to read through the guidance documents to understand the process and procedure. ICH guidelines for clinical trials are also adopted by HSA. And lastly, HSA has also set up an innovation office to provide scientific and regulatory advice to researchers, academic, biotech, and pharmaceutical companies with an interest in early stage clinical product development and with the intent to pursue product registration in Singapore. Companies can make use of this platform to engage the regulator early on regulatory requirements and seek early guidance on technical or scientific issues that need to be considered during product development, including non-clinical development, clinical development, quality development such as CMC, manufacturing and good manufacturing practices, GMP, and regulatory submission. The Innovation Office also accepts requests to meet. And you do need to provide at least 2 weeks prior to the meeting of the following information: description of your company or institution, description of the product, specific question to be addressed, and briefly information relevant to the question raised. And now we will have our last poll of the day. Poll, please. So in Singapore, for product registration of Class 2 cell, tissue, or gene therapy, how many evaluation route and application routes are there? This is a bit tricky. Okay, I'll pull up some hints on my slides.
Can you see the results, Wei Xia?
I can't.
Oh, okay.
Have you released the results? Okay, yeah.
Yeah, okay.
I saw the results. Yeah, so majority of you got it right. Answer is 2 and 3. So there's 2 evaluation route, which is full or abridged, depending on the status of approval by comparable overseas regulators at the time of submission, and 3 different types of applications, which is the NDA 1, 2, and 3. So I come to the end of today's presentation. Thank you everyone for listening.
Perfect timing, Wei Xia. Thank you very much. We have 15 minutes in which to cover off any questions. And there were two questions from Gary. I'll give you the first one, which was, how would you recommend defining the starting materials for a autologous therapy? Is this the PBMC or leukapheresis material?
Actually, I would think that it will be the leukapheresis material, but the problem is you have to define it and tell and write it in your dossier which starting material that the companies deem it as the starting material. So depending of the product, what is the biological properties of the product, either one of it can be the starting material, but you do need to justify and write it in your dossier how you define it and how you define which of each is the starting materials.
Okay, thank you. Gary, does that answer the question? Or you have any comment? Not sure if you can unmute. Okay, no, he said that's fine. Okay, I'm going to ask the second question, which Pei Ching poses. For autologous products, is comparability study performed using surrogate T cells or cell lines or real patient-derived materials?
The best would be using a real patient-derived material. Authority do understand that it's very hard to use real patient-derived material. So other kind of materials is acceptable. I would prefer using healthy donor. I mean, based on my experience with HSA, healthy donors will be a more comparable materials to be used to test in the comparability studies. But of course, like I mentioned, the best is to use a patient patient cells to do for comparability test, but it might be hard to do that, and it's a waste of precious resources or cells to be used on comparability study. So whichever cell lines or whichever cell material you are using as a comparability study, you have to justify and provide justification to the authority and state it clearly to why this is the most appropriate cells to be used for the comparability studies.
Thank you. Pei Ching, was that okay?
Yeah, absolutely. Thanks, Wei Xia. Actually, I would like to extend a little bit and just to ask a similar question in terms of reference standard. So can I say the same logic or same principle also applies to reference standard?
Yes, you can. So basically, whichever reference standard material that you're going to use as a reference standard, you do have to provide justification and rationale and state it very clearly in your dossier why the company or the drug manufacturer deemed this as the best reference standard to be used.
Got it. Thank you, Wei Xia.
Okay, the second question from Gary was, if CTs are manufactured outside of Singapore, is a distribution license required for importation into Singapore?
Sorry, what's CT?
Cell therapies, sorry.
Cell therapy. Sorry, you mean if cell therapy are manufactured overseas?
Yes, that's right.
And then are you intending to import into Singapore or?
Yes, of course, if it's meant to treat within Singapore, I mean, under HSA, do you require a separate distribution license like traditional pharmaceuticals?
Yes, so you do have to so if you're intending to sell it, I mean, as a commercialized product, is that what you mean?
Yes.
Are you intending to do it as a commercialized product or
I guess there's two things, right? One, if it's not a commercial product, you will be covered under the CTA, right, route. So you can bring that in under CTA route, but because it's a commercial product, I just want to confirm that then a distribution license is required after approval as well.
After approval as a commercialized product. Sorry,
Correct.
okay.
That's right.
for that, you need. So you first need to register the product in Singapore, and then you have to supply all the information in your quality dossier, and also the information of the manufacturer facilities. And then upon approval, then there might or might not be a need for the regulator to go to the site to review. Of course, if the manufacturer facilities overseas has any license, any documents, you can submit it as well during your applications. Because
Sure.
our authority might need to audit the overseas sites as well. So that will be depending on the risk assessment by the authority.
Thank you for that question, Gary, and also for answering that, Wei Xia. Are there any other questions? Please feel free to unmute yourself if you'd like to ask personally, or else just put it in the chat.
But for any question you have regarding HSA, feel free to contact them because they are very friendly and helpful. Yeah.
Always good to know, right?
Yes, they are very friendly and helpful in answering the questions.
Yeah, that's great.
So especially I do highly recommend for any SME or any if there's any institutes, local institutes, academic institutes that has an interest in going to the CTGDP product, contact Innovation Office for their help if you are not familiar with the regulatory.
Good advice.
Yeah.
Okay, so this is your last chance to ask a question. Otherwise, we will take leave and end a little bit early. Okay, no other questions. So I'd like to thank Wei Xia for your time and for sharing a very comprehensive and informative presentation this evening, and to thank everyone for joining us on this afternoon. If you missed any of it and you were not able to catch the beginning, please go to our ISPE Singapore YouTube page. It will be posted up later on, I think in the week or early next week. So, and there are lots of other presentations, Technical Tuesdays, conference presentations that are also on there. So please do use that as a resource. So thank you very much for your time, Wei Xia.
Thank you.
It's been great to have you join us this evening. And thanks everyone for joining. We look forward to seeing you all again soon. And if you have any questions for Wei Xia, please feel free to email ISPE Singapore, which is mostly Christina, who's been emailing all of you, and we will forward the questions on. Okay, so with that, I'd like to bid everyone a lovely evening, and thanks all for making it. Take care, everyone.
Thanks.
Bye.
Bye.
Bye.